All right, for once I am getting 2 videos done in 2 days, so be impressed, guys. We're going to talk about cell division now and the cell cycle. So very quickly, before we get into the more complex bits of the cell cycle that are involved in eukaryotic cell reproduction, let's talk about asexual reproduction real quick. So asexual reproduction of cells produces genetic clones. There's no variation. There is a much simpler process for asexual reproduction in unicellular creatures, mostly because a lot of these tend to be prokaryotic. They don't have a nucleus, so it's a lot easier for them to divide. What they do first is they make a copy of their DNA. So this lovely little tangle that you see here is a chromosome, a single bacterial chromosome. They anchor it at a point in the cell membrane, they reproduce it, and then basically just grow the cell in between the places where you have these chromosomes anchored. Once they are separated enough, you start forming this kind of, you start pinching in between the 2 different chromosomes until eventually they have separated and you have 2 new cells formed. This process in prokaryotes is called binary fission, literally 2 splitting into. Binary meaning 2, fission meaning splitting. This process is more complicated in eukaryotes, where the process of nuclear division is called mitosis. So there are some differences between prokaryotic cells and eukaryotic cells that complicate the matter. Bacteria only have a single chromosome. It is circular. It is much more difficult to tangle up, and it is fairly simple. However, in eukaryotic cells, you have much more complicated, much longer linear chromosomes, and you have more than one of them. So you've got to keep track of all of those chromosomes and make sure that you get 1 copy of each chromosome into each cell that's being formed. These are replicated during a part of the cell cycle called S phase. The S stands for synthesis, and they are distributed during mitosis. By the way, we're going to talk about chromosomes and how they condense. We'll talk in more detail about the structure of DNA when we get to our DNA unit next, but chromosomes are basically wound up DNA. So this is the double helix that you normally think of. This DNA strand is wound around these proteins called histones. Those groups of 4, or actually it's 8 histones with DNA wrapped around it form something called a nucleosome, which then coils up, and then those coils coil up and eventually form this more compact structure called a chromosome. This, by the way, chromosome is made up of 2 identical sister chromatids that are attached to each other. Sister chromatids are identical copies of a chromosome. So they are 100% identical. They are going to be separated during mitosis. So you only really see these X-shaped structures that we think of as chromosomes right before cell division is going to happen. However, cell division is only part of the larger cell cycle. The cell cycle has a very important purpose to make new cells. This is needed if you are a single-celled creature like this yeast cell right here, which is a fungus, and you need to reproduce a new organism. It is important if you are trying to grow. So this is the tip of the root of a plant, and we're going to do a lab next week where we're going to look at mitosis happening in the tip of root cells. In root cells at the tip, and then you also need it to repair injuries. So if you get a cut or something like that and you produce new cells to fix it, that is going to be due to mitosis. However, you don't want uncontrolled cell growth because that leads to its own problems, which we'll talk about later. This diagram is one that you got in class, so you can fill in some of the notes that we have during class. This is just showing you the entirety of the cell cycle. So a lot of times you'll start looking at the cell cycle here at G1, also called gap 1. That is where a cell grows after division. This is also where a cell performs a lot of its normal functions. This star that you see here is something that we're going to talk about. It's a cell cycle checkpoint, and that is when a cell is going to check, hey, is it time to divide? Should I divide, or should I exit the cell cycle and go to something called G0? gap 0, which is where a cell is no longer dividing. So there are some populations of cells in your body that at a certain point will stop dividing. They exit the cell cycle permanently. There are others that will go into G0 until they get a signal saying, hey, we need more of your cell type, and then they will re-enter the cell cycle and go ahead and start reproducing again. Notice that I have this diagram in the center that is showing you a stylized version of the cell, so you can see relative changes in sizes, and also importantly, you can see when chromosomes are reproduced. So this is a single unduplicated chromosome, whereas during S phase, you'll notice we have 2 identical chromosomes that are now joined together. Gap 2 phase, or G2, is where you're growing even larger because you're about to enter mitosis, and then this checkpoint right here is where you the cell will decide, is everything okay? Have we duplicated all of our DNA? Are we ready to move on to the next phase and divide up our DNA? Then mitosis is the stage of nuclear division. We have a checkpoint at this point where the cell will stop and check, am I ready to divide up my DNA properly? At any of these checkpoints, if there is an error or if the cell decides no, it's not ready to move on to the next stage, it pauses. It can either fix the problem, or if the problem is beyond fixing, let's say there's some catastrophic error with DNA replication and you just don't have enough chromosomes and everything's mixed up, then that cell can actually undergo apoptosis to make sure that you don't have a mutated, messed up cell running around. After mitosis, you have the division of cytoplasm, which is called cytokinesis, and that produces a brand new daughter cell. So very quickly, let's look at how chromosomes are structured. You have a centromere that connects the 2 sister chromatids. And let's talk about the centromere in a little more detail. So the centromere is important not just to attach 2 sister chromatids to each other. It's also important because it attaches to what are called spindle fibers. These are microtubules that are basically going to feed through the kinetochore here. And as they feed through, they're broken down. And then these kind of motor proteins are going to basically work their way up the microtubule, and they're going to yank the chromosome along behind them. So if you don't have kinetochores perfectly attached to each side of the centromere, then you won't be able to go on with mitosis. And they are motor proteins, and this all relates back to when we were talking about the cytoskeleton in the previous lesson. All right, so aside from the chromosomes, there are a couple of other important structures. The centrioles, which are made up of groups of microtubules. They are centrioles or centrosomes are the bodies that are found in animal cells. In plant cells, they don't have these distinct centrioles. Instead, they have an MTOC, or microtubule organizing center, and its purpose is to form this structure right here called the protein spindle. The protein spindle are these microtubules that are going to go and attach to the centromeres of chromosomes. They're going to shorten, and they're going to pull those centromeres and the associated chromosomes to opposite ends of the cell. So in this diagram, you can actually see the microtubules, which are these orange dots right here, and the green coming out, those are the fibers of the protein spindle. So mitosis is the process of nuclear division. Remember that it's followed by the process of cytoplasmic division, or cytokinesis, but since we have multiple chromosomes that each carry different and distinct genes, this process is kind of like a fancy, very well-orchestrated dance of moving your chromosomes around to make sure each daughter cell is going to get a full copy of the genome. So in biology, in your freshman year, you may have memorized all the names of the phases. You do not need to do that for AP Bio. Instead, what you need to know is that there are these events that happen in mitosis. So at a certain point, you have kinetochores attached to the centromeres, and that is attaching these microtubules, which are parts of the protein spindle. In fact, the mitotic checkpoint checks for this, and at the mitotic checkpoint, you pause. If you don't have microtubules attached to each kinetochore, then you pause, you make sure they get attached, and if you can't attach all of your kinetochores to microtubules, you stop mitosis. Then the next important part is alignment. You're going to line up those chromosomes at the center of the cell, so that way you can pull them to each side. And then finally, you have the separation where you are pulling these chromosomes to opposite ends of the cell. That way, each new cell, when it separates somewhere in between these, like right here, is going to have a full complement of chromosomes, and this is done by basically the kinetochores pulling themselves up along microtubules and the microtubules being broken down behind them. Cytokinesis then follows. It's different in animal cells versus plant cells. What happens in animal cells is you have in plant cells is you have vesicles line up along the center between the two newly forming nuclei. They're going to contain elements of cell wall. They're going to join with each other, and then when they join up at the edges of the existing cell wall, they will have formed two new cells. On the other hand, animal cells have a much simpler process since they don't have a cell wall to build. All they do is basically a ring of microtubules around the center of the cell starts contracting. I think of it almost like a very chunky person with a belt on that's too tight. Well, in the case of animal cells, during this process of what's called cleavage, it's like you took that belt around a chunky person and you just kept squeezing it tighter and tighter until that person popped apart into two different people. Luckily, that doesn't happen to people, but when it happens to cells, they are able to form two new cells. And this does happen concurrently with mitosis. So while mitosis is finishing up, cytokinesis is going to go ahead and start. Now, I've mentioned checkpoints a couple of times. These checkpoints are going to regulate progression through the cell cycle. And at each of these checkpoints, you're going to pause and say, okay, is everything okay in the cell? Can we move on to the next stage of the cell cycle? So there are several different checkpoints. The G1 checkpoint, also called a restriction point, is basically going to be influenced in a lot of cases by external factors. So are there growth factors nearby that are telling us, hey, we need to produce more cells? Are there sufficient nutrients that we can support new cells? And is our DNA all right? Or do we have any mutations, DNA damage? If we have DNA damage, that could be enough to prompt apoptosis. However, if we don't have enough resources or there are no growth factors, then the cell cycle can be paused and it can be left in G0. G2 is going to be asking the questions, do we have enough organelles? Do we have enough proteins? If we don't, we need to pause the cell cycle and make more. Do we have correct and proper replication of each of our chromosomes? You can go back and check for error in the DNA, and once again you can pause the cell cycle to mediate any of these issues. And finally, the M checkpoint is basically, do you have your chromosomes attached to your spindle? This is to make sure that your chromosomes divide up properly, because if they don't, if you have a cell with a missing chromosome, it probably won't be able to function and it won't be able to survive. So until you have all those pairs of sister chromatids attached to these spindle fibers from opposite sides of the cell, you will not progress through the rest of mitosis. These checkpoints are moderated by groups of proteins called cyclins and cyclin-dependent kinases, or CDKs. So CDKs are enzymes, and their job is to add a phosphate group onto other proteins. When they do that, they will turn on that protein or turn off that protein. So CDKs are inactive until they bind to a cyclin. So right here, this is an inactive CDK. It's not going to do anything, it's just sitting there. When a cyclin, the appropriate cyclin, comes along and binds to it, then that activates it, and now this CDK can go on and do its job, act as a signal to, say, move on to the next part of the cell cycle. CDKs tend to stick around in a cell long term, but cyclins tend to be made and degraded at different points in the cell cycle. So these turn out to be the on-off switches, and your cells will produce cyclins if it's possible to move on to the next part of the cell cycle. So cell cycle progression depends on making and then breaking down these cyclins. So here we have a cyclin that's involved in the G1 checkpoint. It's produced at the beginning of G1. It builds up to high enough levels, binds to the CDK, activates it, and then that acts as a signal to move on to the next part of the cell cycle, after which that cyclin is degraded and broken down, and that CDK sticks around until the next cell cycle when we're approaching a G1 checkpoint, and if enough cyclin is produced, then that CDK can be turned on and then keep the cell progressing through the cell cycle. This is just another way of showing you that. So when cyclin and CDK bind to each other, then CDK is active, it becomes a kinase, and it is able to add phosphates onto other proteins. That target protein can then go on to do another job. I do have a video that I'll put the link to in the description that is moderately complicated because cyclins and CDKs can get a little complex, but it's also pretty good at showing how cyclins and CDKs actually control the cell cycle. So there is an important cell cycle CDK/cyclin compound. It's called mitosis promoting factor, or MPF. It is involved in the M checkpoint, and so when CDK and cyclin bind to each other and form this MPF, it's going to initiate mitosis, and then it's degraded. The cyclin MPF breaks apart and is no longer able to signal, hey, let's go into mitosis. Growth factors are important because what they'll do is they will stimulate growth of cells, so they will actually initiate mitosis. A lot of these are going to end in GF, or growth factor, as part of their name. So there's NGF, which is nerve growth factor, there's TGF beta, which is transforming growth factor beta, and there's PDGF, or platelet derived growth factor. Each of these growth factors are turned on at different times in the cell cycle and may act on different cell populations. Then you have this important class of proteins called tumor suppressor genes. Their job is to make sure that if there is DNA damage, if there's any problem, that the cell cycle will stop. And in some cases, this guy, for example, P53, is a very important tumor suppressor gene. When it's activated, it can not only pause the cell cycle, if damage is sufficient, then it can actually initiate apoptosis. The cell dies, and then you don't have a problem with that cell anymore. When you don't have P53 active, when it's damaged in some way or the gene is mutated, it no longer stops the cell cycle. There's no longer control over the cell cycle, even if there is mutations. And so the cell starts growing out of control. It starts creating a population of other cells that will now have a non-functioning tumor suppressor gene, and then that cell can become cancerous and can form a tumor, and that can cause a problem. These genes are named tumor suppressor genes because they were first discovered in association with cancers. When these genes were not working, you had tumors. When these genes were working, those tumors were suppressed. And then, like I said, there are some cells that exit the cell cycle and end up living most of our lives in G0. Most neurons in the body, as well as a lot of muscle cells, are going to stay in G0. And in fact, a lot of medical research focuses on, can we get some of these cells to re-enter the cell cycle and divide, especially in cases, for example, like with neurons, if we have nerve damage, a stroke, or any damage to the spinal cord, can we get neurogenesis, or the creation of more nerve cells, to occur? All right, that is your very short lesson on the cell cycle and mitosis. Look in the description for any video links that I have that might be helpful for you.